Good afternoon. The title of my dissertation talk is Grey Matter Surface Based Spatial Statistics in Neuroimaging Studies. In this talk, I'll go over the main motivation behind this study and then the corresponding challenges in approaching such study, followed by the research goals and then contributions that can be used as building blocks for performing group studies in neuroimaging. So brain related disorders are affecting millions of people across the world. And just within USA, these are the top brain related disorders or like Alzheimer's, psychiatric disorders, etc. These can be manifested into microstructural changes. For example, here is the microcircuit model for schizophrenia, where ex vivo studies have shown reduced dendritic spines in schizophrenic population compared to that of the healthy controls. So there is an opportunity here to capture this kind of information with neuroimaging. For example, here are the brain volume changes that are captured using neuroimaging that shows different regions of brain changing differently with respect to that of age. Let's look at the anatomy of interest that we'll be using in the rest of the talk. So here is the picture of the brain where the inside region is called white matter and the outside region that we see here surrounding the brain is called gray matter. So that is key to many executive functions of the brain. And the central region that we see here is called cerebrospinal fluid. So this information can be captured from structural T1 image, and this can also be used for reconstructing a cortical surface, which is a 2D mesh representation of the surrounding area of the brain. So this can capture both sulcus as well as gyrus, along which we can derive the matrix of interest. So these regions could be further divided into multiple anatomical regions, into smaller anatomical regions based on the clinical study of interest. And then we can also acquire diffusion MRI that can provide microstructural indices. For example, the zoomed in box here shows the axonal pathways going from gray matter to white matter and vice versa. So this kind of information can be captured using fiber tractography, which is a diffusion tensor imaging model. So basically diffusion MRI captures the diffusion of water molecules within the brain, from which the microstructure information can be reconstructed using any specific model like diffusion tensor MRI, diffusion tensor models, etc. So thanks to the BRAIN initiative, we now have increasing number of neuroimaging databases, and this gives us an opportunity to test hypotheses of both developmental growth as well as disease conditions. Okay, so this is great, right? So we have large set of data images that are available across multiple modalities. We can combine that with demographic and clinical information and then put that into some magic learning model, and we should be able to capture outcomes like diagnosis, brain age, etc., right? But things are not always this simple, right? So we do have challenges involved when we are dealing with multimodal images in neuroimaging studies, especially when we want to understand changes in brain microstructure. So what are those challenges and how we can improve statistical power so that we can predict clinical outcomes? So this is the main motivation behind this study. So let's look at the challenges first. So there are a number of steps involved when we are quantifying the brain signal to conduct group studies. First, we acquire the MRI and get corresponding image like T1 weighted diffusion MRI, etc. And then we derive corresponding metrics of interest based on the underlying model. For example, we get segmentations from the T1 weighted image, and then we get advanced diffusion MRI measures from the diffusion weighted imaging. Once we have this information, we align all this onto the template space so that we can perform group analysis. So there are a lot of steps here, and each step has potential to introduce error. If we zoom out and see, we have the scan protocol selection happening at the first step. And once we have the data acquired in multiple modalities, we need to make sure the intra-subject data alignment happens so that we can analyze data across multiple modalities. And when we are dealing with the group analysis, we also want to make sure how reproducible a given model is. And finally, when we are aligning data onto the target surface template, the challenge could be when we have alignment issues as well as if there is a bias in the template selection. So this is a vast research area, but the main focus would be in the last two blocks. When I say alignment issue, here is an example where we do have well matched images with volume deformation, whereas when we look at the right hand side, the zoomed in region, we do see that the cortical structure is still not well matched between these two subjects. So this is the kind of challenge that we'll be looking into on how we can do group analysis in gray matter across these multiple subjects by overcoming these challenges. Okay, so now we have all the data moved onto the template space from multiple subjects across multiple modalities. So one issue that we could run into at this point is when we have site or scanner related differences. We have to make sure that there are no site or scanner related differences before we can perform group analysis. So there are multiple ways in which group analysis can be performed. It could be region of interest based analysis, or it can be voxel based morphometry analysis, or it can be voxel or vertex level analysis. Each comes its own pros and cons, but voxel or vertex level analysis has higher statistical power and high spatial and spatial sensitivity and specificity compared to the other two models. So the main focus for our study would be to do the data harmonization first and then also address spatial statistics in gray matter so that we can perform non-parametric tests in gray matter. Okay, so now the recent trend has been to use deep learning because it has been become a popular model due to the higher computational power that we have gained as well as the availability of large scale data sets, especially in the neuroimaging domain. So here is the meta review that was submitted to neuroscience and biobehavioral reviews, where the blue bar here shows the deep learning model and red bar shows the kernel based approaches. So in almost all these studies which have taken across multiple clinical studies like ADHD, Alzheimer's, etc., deep learning models are having higher accuracy compared to that of the kernel based approaches. Deep learning model has also been shown to perform semantic segmentation and it has seen success in volume domain. For example, here is the brain segmentation that is performed using a deep learning model, and this work is published in MICAI 2018. So while this has seen success in the volume domain because of the spatial grid structure, this is still a challenge area for the cortical surfaces to perform semantic segmentation because we do have a 2D mesh representation which is not a spatial grid or it is not a structural grid information that is used in the traditional deep learning approaches. So this is one challenge area that we'll look into on if we can do deep learning on cortical surfaces for doing the semantic segmentation. Okay, so to summarize the research goals, we would like to improve statistical power in quantifying brain signal, especially in gray matter for group studies. So to do that, our first task would be to improve spatial statistics in gray matter, and then for addressing the source of bias in group studies, we will look into these three areas of model reproducibility, data harmonization, and template bias. And finally, we apply the deep learning models onto the cortical surfaces. So for this study, we are looking into NODI model, that is the model of our interest. It is called neurite orientation dispersion and density imaging. Basically, it's a model-based technique from diffusion MRI that can unentangle compartments into intracellular and extracellular components. So for example, here is the NODI model where it can separate out the isotropic volume fraction first, and from whatever is remaining, it will divide that into intracellular volume fraction and extracellular volume fraction. So within the associated with intracellular volume fraction, we also get additional metric called orientation dispersion index. So basically what this provides is it shows how dispersed each of the neurites are within a given voxel. So all we need to remember from here is that ODI is high in gray matter and it is low in white matter. Okay, so let's look at the contributions. So here are the 5 contributions, where first we'll be looking into spatial statistics in gray matter for doing spatial statistics in low resolution images and show applications in diffusion MRI and functional MRI. For model reproducibility, we'll be looking into empirical reproducibility of NODI model, and then for doing the data harmonization, we want to address site or scanner related differences. We want to correct for those differences and see if we can gain statistical power. And for the template bias, we show an approach how we can construct an unbiased cortical surface template in feature space. And finally, we apply the deep learning models on cortical surfaces with an application of semantic segmentation showing auto labeling of sulcal curves on the cortical surface. So let's look at the first contribution, which is spatial statistics in gray matter for low resolution images. So why is this important? Gray matter cerebral cortex is less than 5 millimeters thick, but it is key to many executive functions of the brain. While structural MRI can be acquired at high resolution, the images in other modalities are acquired at much lower resolution because of time constraints. So this can lead into challenges like registration artifacts and partial volume effects when we are doing group studies. In order to overcome these kind of challenges, it was shown in white matter by using track-based spatial statistics, where they proposed a projection onto the alignment invariant track representation. That is the mean FA skeleton, as we see here on the green. So they derive the mean FA skeleton and then project all the metrics of interest onto this mean FA skeleton for doing the cross-subject analysis on this target skeleton. Similar approach has been adapted, and gray matter based skeleton approach is shown here. Instead of using the white matter skeleton, here they show gray matter skeleton and then project all the information onto the gray matter skeleton for performing group studies. But the problem with this skeleton based approach is that it's less sensitive, as it's not a true representation of the cortical structure. So in order to overcome that, we show surface-based approach where we take advantage of high-resolution T1 image and then get its corresponding cortical surface and then apply the volume deformation that we have from the volume image onto the surface so that we now have the surface in volume domain. But as I've shown in the previous example, even though the volume information is well aligned, we still see some discrepancy in the cortical structure between the subjects. In order to overcome that, we want to project all this information onto a single target surface. So that means we want to get mapping from this yellow surface onto the blue surface that we are seeing here. In order to do that, we make use of the surface registration step to obtain the corresponding mapping. So once we have the mapping, we can take the co-registered images from the low resolution diffusion MRI, etc., and derive the corresponding metrics of interest. Here I am showing NODI metrics, and then transfer them onto the cortical surface of the subject. So once we have this information on the surface, we can project them onto the target surface using the mappings that we obtained in the above step. So here are the differences where we do see improved representation of cortical structure with this yellow surface, that is the cortical surface, compared to that of the skeleton based approach, which is the blue skeleton that we see around the brain here. And it can yield more number of highly probable gray matter voxels. So for the experimental data, we looked into this hypothesis where it is shown that schizophrenic population has reduced dendritic spines compared to that of the healthy controls. In order to test this hypothesis, we took orientation dispersion index, and for the data we have 47 healthy subjects and 67 psychosis patients, which includes both schizophrenia as well as bipolar disorder. And each of these subjects have scan protocol with one structural T1 and two shells of diffusion MRI with two B values that will be used for deriving NODI related metrics. So here are the results where we are seeing reduced orientation dispersion index in medial prefrontal cortex using GSBSS approach. So here we are showing that the psychosis has lower ODI compared to that of the healthy subjects after applying multiple comparison corrections. Here is the significant results that are showing up to be the group differences between healthy and psychosis population. And also this is confirming with the postmortem studies with reduced dendritic length. This is also compared with skeleton based approach, and we do see higher sensitivity in capturing the group differences with GSBSS approach, where it is also able to see differences in medial frontal cortex, whereas it's not captured in the skeleton based approach. And these results were also shown using voxel-wise analysis in the biological parametric mapping, and then compared that with of the larger mask of frontal lobe and shown that we do have lower ODI in the psychosis population compared to that of the healthy controls. And the green that we see here is the gray matter surface, and the blue is the frontal lobe mask that we have applied. And these are published in Biological Psychiatry. Okay, so one assumption that we made here when doing all this mapping is that we do have good co-registration between structural T1 and diffusion MRI. So this may not be always the case, and we could run into issues with alignment in the with correlation. So in that case, the approach that we are taking could have a problem because we are relying on the cortical surface related mapping to put metrics of interest from other modalities onto this cortical surface. In order to overcome that, we make use of the fact that ODI is high in gray matter. Remember, I was showing this image where ODI is high in gray matter and low in white matter. So instead of taking the enclosing voxel approach, that is, instead of taking the metrics of interest from the enclosing voxel to project onto the surface, we now take vertex normal search to fetch highly probable gray matter voxel by making use of high ODI. So basically we look along the vertex normal and pick the highest ODI voxel and only take the metric from that voxel and project it onto the surface. As we see here on the right hand side, we see that after ODI search, we do see higher ODI than the before search ODI. Okay, so let's look at the complete workflow now. So this is the information that we have seen before. For obtaining the mapping using high resolution T1 image, basically we get the cortical surface, do the volume registration, and then perform the surface registration step to get the mapping. Now for the low resolution images, if we have some co-registration issue, we are now performing cortical ODI search, and then in addition to diffusion MRI, we are now also showing this for functional MRI data. We can also apply this for functional MRI data where it will be similar to having first level analysis to get corresponding metrics of interest and then transfer them all onto the their subject specific surface. So once we have this information on this subject specific surface, we can then apply the GSPSS mapping and perform spatial statistics of these multimodal features on a single target surface. Here are the results of 30 healthy subjects for mean ODI value. And on the left hand side, we are seeing GSPSS with no search, and also in the middle we have GSPSS with 2 mm search. These results are compared with that of the SIFTIFY pipeline, which is adapted from Human Connectome Minimum Processing pipeline for non-HCP datasets. So as we see here, we do see higher ODI across the multiple subjects, across the 30 healthy subjects compared to that of the without search, indicating we are overcoming the partial volume effects that are coming from the lower ODI from white matter region. And also the other interesting observation that we made here is we may lose some cortical structure related information, and as we are only seeking highly probable gray matter voxels, whereas we have some cortical structure showing up in SIFTIFY or the without search kind of approach, whereas we may lose that cortical structure related information with high ODI search. And we now showed another application in functional fMRI. So by comparing 3 working memory fMRI tasks. So basically here we compared GSPSS with volume based registration, surface based registration, and also the SIFTIFY pipeline. And on the last 2 rows we are showing GSPSS without search and with search. So the higher the activation, the better, because if there is an increase in neural activity, there will be a small amount of increase in the MR signal. So this is what is getting captured in functional fMRI. So this is the indirect effect of increase in blood flow. So if we have an increase in neural activity, there will be increase in blood flow, and that's what is getting captured as the increase in the MR signal. So this is the quantitative comparison of the percentage activation, and we see that GSPSS with search has higher activation compared to any of the other methods in all the 3 tasks. So while we showed that statistical power improved in both experiments of diffusion MRI and functional MRI, we do not have a ground truth. So in order to check for sensitivity and specificity of this method, we have performed the simulation study where we took gray matter density and then of 30 healthy subjects and then divided them into two groups and looked at the difference between the two groups and took it as a baseline. Then we took this three ROIs with 3 millimeters, 4 millimeters, and 5 millimeters size, and then changed the intensity within the ROI in a single group by 10%, 20%, up to 90% in the intervals of 10. And then we looked at the group differences again between those two groups. So one group is unchanged. For the second group, we changed the intensity value within a specific ROI and then compared the group differences again. So now what we are showing here is the maximum t statistics ratio where we are normalizing it by the baseline. So here is the comparison and we see that GSPSS is highly sensitive as well as specific, indicating it can capture the differences between the two groups even with a smaller ROI. So here we applied GSPSS on multiple clinical data sets and show similar dice coefficients across all these data sets. These are the four different lobes that we are showing on the cortical surface. And we see that it is robust with scans from different scanners. On the top row, we are showing 3T Philips scanners, and on the bottom row, we are showing Siemens scanners with 1.5 Tesla as well as 3 Tesla. And for the top row, we have psychosis population and also Baltimore longitudinal study of aging, which includes both Alzheimer's disease and mild cognitive impairment along with healthy controls. And on the bottom row, we show datasets from mindfulness-based stress reduction program on both 1.5T as well as 3T scanners. And open source code for this GSS is available on GitHub under MassiLab. So to summarize, here is what we have shown. So basically we have taken advantage of high resolution T1 image to get the mapping that we can use to transfer information from low resolution images onto the target surface. And then we have also extended this to other low resolution images like functional fMRI tasks. Here are the publications related to this area. Okay, so now moving on to addressing the source of bias with in different contexts. So first one, we'll look into model reproducibility. So for this, we are looking into model reproducibility of NODI model, empirical reproducibility of NODI model. So here we have taken a rich data set of single subject acquired on 5 different shells with 96 directions each and with 11 repeats across 3 sessions. So basically we are testing for different scan acquisition protocols, how the reproducibility is with 2 shells of data for VIC as well as ODI and isotopic volume fraction, and we also checked for single shell ODI across different scan acquisition protocols. So basically we divided each scans into multiple scan acquisition protocols by breaking them into different combinations, and we derived almost around 2400 combinations on which we analyzed this data. We also have a accompanying structural T1 image onto which all the data got aligned to, and that's what we normalized our diffusion weighted information onto. So here are the qualitative results where we are showing intracellular volume fraction, ODI, and in isotopic volume fraction. And on the left hand side, we see the gold standard data. That means gold standard is the one where we combined all the shells with all repeats and then derived the NODI metrics across that data. And then we compared that with different scan acquisition protocols. We fixed the lower B value to be equal to 1000 and changed the upper B value from B equal to 1500 to 3000. Similarly, we've changed the number of directions of lower B value from 16 to 88 in the intervals of 8, and then for the upper B value, we changed the directions from 24 to 96 in the intervals of 8. So here we are just showing few representations with lower B value and low number of directions, and lower B value with high number of directions. Similarly, higher B value on the right hand side. And we see that we have the optimal result in comparison with that of the gold standard, somewhere in between, like B equal to 2000 and 2500 with 32 to 64 directions, where we This is optimal for having reasonable scan acquisition time along with having better results comparable to that of the gold standard. Similarly, we have done the single shell ODI analysis where on the top row I'm showing the qualitative results of the ODI across different B values, and then in the second row we are showing difference between gold standard with single shell ODI, and on the last row this is the difference between gold standard with two shell ODI. As we see here, these two results are pretty comparable, single shell versus two shell data. So here are the quantitative results where we are looking at the change in the intracellular volume fraction, the root mean square error of intracellular volume fraction when compared with that of the gold standard across all different scan acquisition protocols. So we have this B equal to 1500 through 3000, and along the rows we have different changing in the directions, the gradient directions for both lower shell and also the upper shell. So as we see here, VIC is sensitive to the outer B shell value, and also it is less sensitive to gradient directions in the SCC and IC compared to that of CS. So basically we are looking at white matter and grey matter, and within the white matter we are looking at splenium of corpus callosum or SCC and internal capsule and also centrum semiovale. So SCC has higher FA, internal capsule has medium FA, and centrum semiovale has low FA. So those are the three regions that we chose on which we have analyzed the reproducibility. So we see that VIC is less sensitive to gradient directions within SCC and IC compared to that of the CS. And also ODI is sensitive to number of gradient directions, and single shell data is comparable to that of the two shell data set as we have seen in the previous slide. And finally, ODI is also highly dependent on the underlying complexity when we are looking in the white matter ROIs. So depending on the underlying complexity, the ODI value is changing accordingly. So these are the publications related to empirical reproducibility. So now moving on to data harmonization. So for this, we are looking into data harmonization across scanners in both white matter and gray matter. So for this, we take the two studies in white matter with one study with two scanners of same field strength and looked at fractional anisotropy. And then on second study, we again looked at fractional anisotropy in white matter, but with scanners of different field strengths of 1.5 Tesla and also 3 Tesla. And then we looked at gray matter by using orientation dispersion index and studied the differences between two scanners of the same field strength of 3 Tesla. So basically here is the approach where we choose a harmonization model that has shown to be effective for correcting the scanner differences and then applied the correction to check the differences in white matter as well as gray matter, and then finally we validated if we see any improvement in the statistical power after applying the correction. So we have chosen this combat model that has shown that additive and multiplicative effects at voxel level while retaining the biological variation. So basically on the left hand side here, we see the corrected value at each voxel for that site specific to the scan. And alpha V is the mean measure, and X is the covariate, and beta is the regression coefficient. Gamma is the additive effect for that voxel for a given site, and delta is the multiplicative effect for the given voxel at that particular site. So earlier applications of these have shown correction for fractional anisotropy in DTI, cortical thickness in structural MRI, and also functional fMRI. So in this approach, in this particular study, we applied this technique on white matter voxels that we obtained from the tract-based skeleton. So basically we get all the white matter voxels from the TBSS mean skeleton and then perform site or scanner related differences and apply the correction on that one. And for gray matter, we took the ODI measure from NODI and then applied GSBSS as I've shown in the previous slides. That's the GSBSS approach that we applied, and then took all the highly probable gray matter voxels and then corrected for the scanner related differences across all those vertices. So for the first study, we have 417 young adults between 23 to 34 years of age, and each of them have a T1 image and also corresponding diffusion weighted MRI. And then we applied track based spatial statistics to obtain the fractional anisotropy on the mean FA skeleton. And then on the top row, we see before correction, we are showing both negative effects of age in pink as well as scanner effects, which are showing up in yellow. And on the bottom row, we show this is after correction. We see that the negative effects of age are still retained while we have corrected for the scanner related differences. In the second study, we looked at 119 elderly subjects ages between 51 and 94 years, which has a corresponding T1 image and also diffusion weighted image. We do the track based spatial statistics, get the corresponding FA on mean FA skeleton, and then again on the top row here, we are seeing biological effects of age, negative effects of age, and then yellow here shows the scanner related differences before correction. And the bottom row shows the scanner related differences after correction. And the pink region that we see here are additional regions showing age related effects. So just to emphasize, these are the additional voxels that got captured in addition to whatever we are seeing on the top row. These are the additional voxels that got captured, indicating that could be a improved statistical power so that it's now able to capture more number of voxels. And then for the third study, we looked into 30 healthy subjects, ages ranging from 23 to 55 years, and it has a corresponding T1 and diffusion weighted MRI. We applied GSBSS processing on ODI metric, and in pink we see the significant scanner effects after applying the multiple comparison correction using FTR correction, and yellow are the uncorrected scanner differences. So these are overlaid on the uncorrected scanner differences. So as we see on the left hand side, before correction we do see the scanner effects, whereas after correction these scanner effects are reduced. We did not include age related effects here because of the smaller sample size. So in this contribution, we have shown harmonization of white matter and gray matter features in diffusion microstructure and have shown that it is able to improve statistical power or retain the biological variability while applying for the scanner correction. And here are the publications related to this area. Okay, so now moving on to template bias. So here we would like to construct unbiased cortical surface feature-based template that we can use for group studies. So basically we would like to have our template not getting biased towards any particular group. So why is this important? When we have this population, this is the model that was put together by Harrison's et al.'s group, where they are showing multiple levels of analysis from biology to macro level. So when we have the population, they could have regional related differences, or it can have genetics related differences, or they can also have developmental related differences. So when we are capturing this information and trying to come with a representative brain, it should be able to reflect the properties of each of those groups in the underlying population. So in order to do that, we take the covariance based weighted average that is obtained by applying the inverse on the covariance matrix. So basically we first get the covariance matrix and then apply the inverse covariance to get corresponding weights, and then finally get the weighted average using this covariance information. So in order to do that, these are the processing steps where we take the structural T1, get corresponding cortical surface, do the initial alignment, and then project it onto a sphere, and then get corresponding features from each of those surfaces, and then calculate both weighted mean and unweighted mean by making use of the a priori covariance matrix. And finally, we perform qualitative and quantitative analysis by comparing differences between these two weighted and unweighted means. So for this, we have done two experiments. First, we took 21 healthy subjects and then we added one repeat of a single subject for multiple times. We added like 20 repeats of rescan for a single subject. And then we took the mean square error between the differences between these two groups for both weighted and also unweighted mean. and the feature that we used here is sulcal depth. And for the second experiment, we took 18 healthy subjects and 18 schizophrenic subjects and took the healthy control mean as well as schizophrenic mean. And then we fixed the healthy control mean, but we changed the schizophrenic subjects from 1 to 10. And then we fixed the schizophrenic mean and then changed the healthy controls from 1 to 10. And then we captured the relative absolute distance as the absolute difference between group mean versus healthy control mean, subtracted that with group mean minus schizophrenic mean, and this is normalized by the difference between healthy control mean and schizophrenic mean. So here are the results from first experiment, where on the top row we are showing unweighted mean and on the bottom row we are showing weighted mean. When we have no repeats, these two have equal values. But when we have repeats for 10 rescans, then we see that this is the repeated scan that we show that we are adding here. When we add it for 10 times, this is getting closer to that of the repeated scan. And similarly, when we have 20 scans added for of the same subject, then the mean value, the unweighted mean value, is almost approaching close to that of the single subject that got added multiple times. Whereas the weighted mean is not affected by how many ever repeats we add the same subject, but it's able to retain and give us the unbiased mean across all these scenarios. Same thing is captured with the qualitative image here, the plot that's showing up here. Okay, so here's the result from the second experiment, where we are taking the relative absolute distance. In the middle row, we have the weighted mean and unweighted mean. They both are same when we have equal number of healthy control subjects and also schizophrenic subjects. But whereas when we have higher number of schizophrenic subjects, then unweighted mean is getting biased towards the schizophrenic mean. Whereas we have higher number of healthy control subjects, then unweighted mean is getting biased towards healthy control mean, because here if it is more towards the red side, that means it is closer to the schizophrenic mean, and when it is closer to the healthy controls, then it is closer to the when it is closer to the healthy control mean, it means it is on the bluer side, whereas the weighted mean is pretty much constant across all these scenarios. So here we showed covariance based unbiased cortical surface template and showed that it is more stable and less biased. Here are the contributions, publications in this contribution area. So finally, we look into deep learning on cortical surfaces. So for this, we are doing a semantic segmentation on cortical surfaces to improve auto labeling of primary sulcal curves using deep learning. So here are the 8 primary sulcal curves on both left hemisphere and right hemisphere. Human cortical primary sulci are relatively stable landmarks and they exhibit phenotypic variability. Basically, they play a key role in cortical surface registration, brain development and regeneration, and also for understanding morphological variability. But the challenge here is labeling is hard because of the high complexity and also the variability in cortical folding patterns in each of those subjects. So in order to do the deep learning, we took normal population for with 1400 sessions with mean age of 72 years, and those are the training and validation data sets, and each scan acquisition has a structural T1 image acquired on a Philips 3T scanner. So here is the workflow where first we do the pre-processing and then obtain the corresponding features that we need for training the model. These include distance map, parcellation, mean curvature, and spectral features. And we also obtain labels for training. This is obtained using the curve labeling method from Liu et al.'s method from 2010. This is also used as the reference method for comparison. And finally, we convert these images on the spherical domain onto planar maps using polar coordinates. And finally, we train the model and perform post-processing so that we can look in. So here are the results of primary sulcal curves, where on the top row we are showing manual labeling across 21 subjects in both left hemisphere and right hemisphere. Compare that with the predicted labeling on the bottom row. And these are the 8 primary sulcal curves. And on the right hand side, we do see two different methods. Basically, the blue bar here is showing the reference method, that is Liu et al.'s method from 2010, and compared that with the green curve, which is the predicted labeling. And as we see, we do see improvement in almost all the curves, and with significant improvement after applying the multiple comparison correction across these two groups in significantly six curves in right hemisphere and three curves in the left hemisphere. And if we take a closer look, while we have pretty good match between manual labeling and predicted labeling, we do have some discrepancy showing up in this particular label, that is superior frontal sulcus, in both left hemisphere and right hemisphere when we compare with that of the manual labeling. And the same information is reflected here in the sulcal dice coefficient, where we do see lower sulcal dice in both left hemisphere and right hemisphere for this particular label. But still, it is still higher than that of the reference method, but it is lower compared to the other other labels that we are looking at, sulcal curves that we are looking at. And also the another region where we see some discrepancy is superior temporal sulcus, and this is only reflected in the left hemisphere. And similar information we can see from sulcal dice coefficient, where we see lower value in the left hemisphere, but in right hemisphere it is still fine. So in this approach, we showed auto-labeling results of primary sulcal curves using deep learning. And we did show that they are well matched with the manual labeling. And also we have shown improvement with that of the reference method that we used, that is the Liu et al.'s 2010 method. Here are the contributions related to this area. So finally, to summarize, we would like to improve the statistical power in brain signal for doing the group studies in gray matter. For that, we showed how to improve spatial statistics in gray matter by showing an application in diffusion MRI in comparing differences between psychosis and schizophrenic population. And then we also showed an application in fMRI with three different working memory tasks. And then for addressing the source of bias in group studies, we showed model reproducibility by performing empirical reproducibility of NODI and capturing the observations across different scan protocols in different regions. And then we also showed data harmonization, for which we showed the scanner effects between white matter and gray matter between different scanners, and showed that after applying the correction, we are still retaining the biological effects of age and with improved statistical power. So finally, and also we showed template bias in this particular area, where we showed how to construct an unbiased cortical surface template within a group of population. And finally, we showed deep learning models on cortical surfaces by applying this onto semantic segmentation of deriving auto-labeling sulcal curves, 8 primary sulcal curves on the cortical surface. And the open source code for these two contributions is available on GitHub under MassiLab. So with that, I would like to thank the funding sources that made this study possible, and my committee members for their continuous support, and also all the MassiLab members for extending their support in making these studies happen. Thank you.